This is the cell cycle. The cell cycle begins in interphase. Interphase consists of three primary stages: G1, S phase, and G2. G1 is where the cell does most of its growing and forms the proteins and enzymes required in S phase. S phase can be related to the synthesis of the cell, or the copying of the data. S phase is relatively short, but it is essential to the growth of the cell. G2 is the last part of interphase, and in this stage, the cell resumes what it was doing during the G1 stage. You may think of this stage almost as the cleanup crew, putting everything back in order before M phase begins. Now, onto M phase. This starts with prophase. Prophase is when a cell condenses its unpaired chromatin. After they are condensed, they form chromosomes, and the nuclear membrane begins to, or has already, disappeared. Basically, the chromatin condense and form chromosomes. The next stage in M phase is metaphase. In this stage, the spindle fibers attach to the centromeres of the chromosomes. Also, the chromosomes align across the middle of the cell on a flat plane. This is all for metaphase. After metaphase, there is anaphase. In this stage of M phase, the spindle fibers shorten. Soon after, the centromeres split. Now separated chromosomes, called sister chromatids, are pulled along by corresponding centromeres. The cell also begins to split in this stage. Next, we have telophase. In this stage, the two new cells completely split with each other and form completely new, individual nuclear membranes. The chromosomes now uncoil in their respective nucleuses, and the spindle fibers completely disintegrate. The last stage in mitosis is cytokinesis. This is when the new two daughter cells are split apart completely. They are pinched in the middle, and they form two new cells split from each other. These two cells are the same in every aspect, from their DNA and the same quality of chromosomes. This has been the cell cycle.